The 2006 Nobel Prize in Chemistry, awarded solely to Roger D. Kornberg, recognized his profound contributions to elucidating the molecular basis of eukaryotic transcription. This award culminated decades of research focused on understanding the intricate process by which genetic information, encoded in deoxyribonucleic acid, or DNA, is copied into ribonucleic acid, or RNA, in eukaryotic cells. Kornberg's work provided a detailed, almost atomic-level, understanding of the multi-protein complex known as RNA polymerase or POL and its associated factors, revolutionizing the field of molecular biology and providing crucial insights into fundamental cellular processes, disease mechanisms, and potential therapeutic The path to understanding eukaryotic transcription was paved with numerous earlier discoveries. In prokaryotes, the process appeared simpler, with a single RNA polymerase enzyme responsible for transcribing all genes. Scientists had begun to unravel this process in bacterium Escherichia coli during the 1960s and 1970s. Early work by researchers like Francois Jacob and Jacques Monod demonstrated the existence of messenger RNA, or mRNA, a transient intermediate carrying genetic information from DNA to ribosomes, where proteins are synthesized. In 1969, the Nobel Prize in Physiology or Medicine was awarded to Max Delbruck, Alfred Hershey, and Salvador Luria for their discoveries concerning the replication mechanism and the genetic structure of viruses. This groundbreaking work, while virus-focused, created a surge of molecular biology understanding and exploration. However, eukaryotic transcription presented a far more complex picture. Initial studies revealed the existence of 3 distinct RNA polymerases, each responsible for transcribing different classes of genes. RNA polymerase I, or POL-I, transcribes ribosomal RNA, or rRNA, genes, essential components of ribosomes. RNA polymerase, or POL-I, transcribes transfer RNA, or tRNA, genes which are involved in protein synthesis, as well as other small RNA molecules. RNA polymerase, or POL-I, the focus of Kornberg's research, transcribes messenger RNA or mRNA precursors, which encode protein. The challenge lay in deciphering the precise mechanism by which POL-I, a large and complex enzyme, recognizes specific DNA sequences, initiates transcription, elongates the RNA transcript, and terminates the process. Furthermore, eukaryotic DNA is packaged into chromatin, a highly organized structure consisting of DNA and histone proteins. This packaging presents a significant barrier to transcription. A DNA template is less accessible to the polymer. Kornberg's work, initiated in the 1970s at Stanford University, employed a combination of biochemical and structural biology techniques to dissect the POL-I2 transcription machinery. His group meticulously purified POL-I2 and its associated factors from yeast cells Saccharomyces cerevisiae, a model eukaryotic organism. Through careful biochemical analysis, they identified a set of proteins, termed general transcription factors, or GTF, that are essential for transcription initiation. These factors, including T-phylib, T-phid, T-phili, T-phid, and T-phi, assemble at the promoter a specific DNA sequence located upstream of the gene to be transcribed, to form a pre-initiation complex, or the pivotal mer. In Kornberg's research came with the determination of the high-resolution crystal structure of POL-I2 and its complexes with various transcription factors. X-ray crystallography, a technique that involves diffracting X-rays through a crystallized protein, allows scientists to determine the 3-dimensional structure of the molecule at near atomic resolution. Kornberg and his team faced immense technical challenges in crystallizing such a large and complex protein. In 2001, they published the crystal structure of POL-I2 alone, providing a first detailed view of the enzyme's architecture. This structure revealed a complex arrangement of 12 protein subunits, forming a clamp-like structure that grips the DNA template. The structure also revealed the active site of the enzyme, where RNA synthesis occurs. Subsequent structures, published in the following years, showed POL-I2 in complex with various transcription factors, providing insights into the mechanism of transcription initiation, elongation, and termination. The structure of the POL-I2 initiation complex, for example, revealed how T-phid, which contains the tartar-binding protein, or TBP, recognizes and binds to the tartar box, a DNA sequence found in many eukaryotic promoters. This binding event initiates the assembly of the PIC, recruiting other GTFs and POL-I2 to the promoter. T-phi, a multi-subunit factor, possesses DNA helicase activity, which unwinds the DNA double helix, creating a transcription bubble that allows POL-I2 to access the DNA temp. The structure of POL-I2 during elongation revealed how the enzyme moves along the DNA template, adding nucleotides to the growing RNA transcript. The structure also showed how POL-I2 maintains the accuracy of transcription, proofreading the RNA transcript and removing any incorrectly incorporated nucleotides. Kornberg's structural studies also shed light on the role of chromatin in transcription. He and his colleagues showed how chromatin remodeling complexes, which alter the structure of chromatin and histone-modifying enzymes, which modify histone proteins, can facilitate transcription by making the DNA template more accessible to the impact of Kornberg's work has been profound. His structural insights have provided a detailed understanding of the molecular mechanisms underlying eukaryotic transcription, a process fundamental to all life. This knowledge has had a wide range of applications, from understanding the causes of human diseases to developing new therapies. For example, mutations in genes encoding POL2 subunits or transcription factors have been linked to a variety of human diseases, including cancer, developmental disorders, and neurological disorders. Understanding how these mutations affect transcription can provide insights into the pathogenesis of these diseases and potentially lead to the development of new diagnostic and therapeutic strategies. Kornberg's work has also had a significant impact on drug discovery. POL2, a target for several important drugs, including alpha-amanitin, a toxin produced by the death cap mushroom, which inhibits POL2 in lethal tissues. Understanding the structure of POL2 has allowed researchers to develop more selective and effective inhibitors of the enzyme, which could be used to treat viral infections or cancer. The legacy of Kornberg's research extends far beyond the specific details of eukaryotic transcription. His work has demonstrated the power of structural biology in elucidating complex biological processes. His meticulous approach to protein purification, crystallization, and structure determination has set a high standard for the field. He has also trained a generation of scientists who are now making their own contributions to our understanding of the molecular basis of life. It would be negligent to overlook the broader context of scientific recognition. While the Nobel Prize justly lauded Kornberg's accomplishments, it also sparked a degree of controversy. Some researchers felt the prize should have been shared with other scientists who made significant contributions to the field of eukaryotic transcription, particularly those involved in the discovery of transcription factors or the development of techniques used in structural biology. The decision to award the prize solely to Kornberg highlighted the inherent complexities in assigning credit for scientific discoveries, particularly in collaborative fields like molecular biology. One can also appreciate the historical irony associated with the initial resistance to Kornberg's findings. Early models of transcription favored a more simplistic view, with POL2 acting largely independently. Kornberg's demonstration of the crucial role of numerous associated factors, forming a complex and highly regulated machinery, was initially met with skepticism. His persistence, supported by rigorous structural data, ultimately overturned established dogma and ushered in a new era of understanding. In conclusion, Roger D. Kornberg's work on the molecular basis of eukaryotic transcription represents a landmark achievement in molecular biology. His determination of the structure of POL2 and its associated factors provided a detailed, almost atomic level, understanding of this fundamental process. His work has had a profound impact on our understanding of human diseases, drug discovery, and the fundamental mechanisms of life. His legacy continues to inspire scientists to pursue ambitious and challenging questions in biology, employing innovative techniques and pushing the boundaries of our knowledge. His contribution to the understanding of DNA transcription remains paramount, showcasing how meticulous research in structural biology can reveal the intricacies of cellular processes, impacting both fundamental science and medicine with lasting implications.